everyone. Now that we've had a chance to describe the basic structure of the neuron, we want to also describe some of the characteristics of the different types of neuroglia. So there are 4 types of neuroglia in the CNS and 2 types in the PNS, and we're going to cover all of them in this video. So we'll start with the neuroglia that belong to the CNS. The first is astrocytes, and we could think of them as sort of almost being star-shaped with some very long extensions. So just to kind of get the full picture here, we're going to draw 2 neurons, and we can see that the neurons, there's a synapse between the 2 neurons. We have an axon and a dendrite. And then the astrocyte actually, it has a lot of functions, and so we're going to kind of see it extend to a lot of different things. And then we'll be able to sort of just explain all of those different functions. So the first function that we want to mention is that it isolates the synapse. And what that means is when there's communication between the presynaptic neuron and the postsynaptic neuron, the one before the synapse and the one after, the communication is going to it's going to involve neurotransmitters, and we want to make sure those neurotransmitters kind of stay in the synapse and they don't take off kind of elsewhere. So it sort of ensures that the message gets delivered where it's supposed to go. The next function of the astrocyte is to take up ions and neurotransmitters from the ECF and from the synaptic cleft. So some of the neurotransmitters can get taken up by the astrocyte, and then also probably the most important ion that's picked up by the astrocyte is potassium. So potassium, it can actually affect the behavior of the neurons. And so if one astrocyte picks up a whole bunch of potassium, what it will do is sort of redistribute that potassium. There are gap junctions between astrocytes. So a lot of astrocytes are connected. And so when there's an abundance of potassium in one astrocyte, because in a local environment there was an excess, the potassium actually flows through those gap junctions and sort of evens out among many astrocytes. So there's not like an excess accumulation of the potassium in that one astrocyte, and then prevention of picking up more potassium as needed. Now, if there was a major sort of body malfunction, the behavior would be different. There's a different mechanism for taking care of the potassium. So this assumes it's sort of an isolated issue. The next thing we want to mention about astrocytes is their role in the blood-brain barrier. So if we think of the capillaries that feed the brain tissue, the capillaries actually form what's called the blood-brain barrier. There are tight junctions between all of the endothelial cells. So just in case we didn't mention this before, the endothelial cells are the epithelial cells of capillaries, or the epithelial cells of blood vessels. And so in capillaries, it's just one single layer of cells, and we call them endothelial cells. And those cells, sometimes they have tight junctions, but not always. But in the brain, they always do. Now, the astrocytes extend some of their feet to the capillaries as well. So in addition to the blood-brain barrier being maintained by the tight junctions, the astrocytes have the ability to monitor the stuff that's moving out of those capillaries and into the ECF, or to what's going to be able to interact with the neurons. And they can also limit the exchange of certain things like sodium, potassium, and carbon dioxide. So the astrocytes they're kind of making sure that the neurons stay healthy and they don't get exposed to anything that they're not supposed to. They also can help repair damaged neurons, and we aren't going to talk about that so much now, but perhaps a bit later. The second type of neuroglia belonging to the CNS is ependymal cells. So ependymal cells are actually epithelial type cells. They're cuboidal or columnar in shape, and their apical surface has cilia, and in some places it has microvilli. So I just want to talk a little bit about where these ependymal cells are located. If we draw a picture of the brain, so think of like a transverse slice of the brain, we can see that there are we could see 2 ventricles. There are actually 4, but we can see 2 ventricles. And these are sort of like hollow chambers in the brain that contain CSF, or cerebrospinal fluid. There's also what's called the central canal in the spinal cord, so like a narrow passageway through the middle of the spinal cord. is the central canal, and that also contains cerebrospinal fluid. And actually, these things are connected. The ventricles and the central canal are connected to each other. So ependymal cells line the ventricles and the central canal, and their responsibility actually is so in some areas, it's to secrete the cerebrospinal fluid in a specific area called the choroid plexus, which we'll describe in another video. And most of the rest of it is to sort of help the cilia sweep the CSF, so it helps to sort of circulate the CSF between the ventricles and the central canal. And just as an aside, the CSF is responsible for offering some cushion and protection to both the brain and the spinal cord. It also transports some nutrients and gases and waste and other chemicals as well. All right, so the third type of neuroglia belonging to the CNS is oligodendrocytes. So I want to just talk about the formation here of the structure we're going to draw in a minute. So think of we have our cell here, and then say that we pressed on one side of the cell and sort of kept pressing. So similar to like if you're thinking of like a toothpaste tube, and you start at the very bottom of the toothpaste tube and you press to sort of bring all the toothpaste up towards the top of the toothpaste tube, and then what you're left with is a section where you have the two sides of the toothpaste tube that sort of have come together, and they're just sort of touching each other, and there's no significant amount of toothpaste in between. So this is what happens with oligodendrocytes. The membrane actually flattens like a pancake in one area and squeezes all the cytoplasm to like another area. And what we're left with is a double layer of lipid membrane. So remember that phospholipid bilayer. If we think about that, we have sort of two layers of phospholipid bilayer. And then we're going to draw a sectional view, like a cross-section of an axon. And that part where the membrane is kind of by itself and all the cytoplasm has been squeezed out, wraps around the axon. So now we'll just sort of take a look at it from like a more of an exterior view. And we can see that the oligodendrocytes actually extend, one cell can extend multiple little flattened sections around multiple axons. So it only wraps around the axon, doesn't wrap around any other part of the neuron. And once it's wrapped around, we actually call it myelin or a myelin sheath. And we'll explain a little bit more about its function when we talk about propagation of action potential. The last type of neuroglia in the CNS is microglia. These are basically resident macrophages. That means that they are a version of white blood cells that perform phagocytosis. So they sort of monitor the environment, and if they encounter a toxin, they just sort of eat that toxin right up. Or if there is a dead neuron or something like that, it could eat that up as well. So it just sort of like mops up the mess that is created by anything bad, cell debris, cells that are dying, or anything else that doesn't belong in the CNS. All right, so just to recap quickly, since I didn't write this before, the CNS contains astrocytes, ependymal cells, oligodendrocytes, and microglia. And the peripheral nervous system actually contains a different set of neuroglia, but there are some similarities. So in the PNS, satellite cells are comparable to astrocytes, and Schwann cells are comparable to oligodendrocytes. So let's take a look at the satellite cells first. So they encapsulate the dorsal root ganglia and the cranial nerve ganglia. I know this doesn't really mean anything yet, but it's a fancy way of saying a bunch of cell bodies in the peripheral nervous system. So basically, the satellite cells sort of cover the cell body of the peripheral neuron, and their job is to regulate the cellular environment in a similar way that the astrocytes regulate the environment in the CNS. And then the Schwann cells, comparable to the oligodendrocytes, so we'll just draw along a neuron with a very long axon. So I don't want to say we're exaggerating because the axon can be extremely long, but in comparison to what we've drawn before, it's a bit longer. So Schwann cells, same sort of explanation in terms of squeezing out the cytoplasm, but unlike oligodendrocytes, we actually only have one Schwann cell can wrap around one axon. It's not possible for the Schwann cell to wrap around multiple axons in order to form myelin. And so when it does actually we actually can see a bunch of Schwann cells wrapped around this axon. And so we get, what we get is a myelinated axon in the peripheral nervous system. Now, it is also fair to say that Schwann cells can interact with unmyelinated axons too. We're not going to talk too much about that, but just for clarity, an unmyelinated axon is not necessarily totally bare. It's just not wrapped in multiple layers of membrane to create the insulation associated with the myelin sheath. All right, so that actually concludes our explanation of the neuroglia. And as we move through the different components of action potential, we'll be able to see some of these neuroglia at work. And in other cases, we might see them at work later on in our long explanation of the nervous system. So I hope you enjoyed it. Thanks for watching.